Am I audible?
Yeah.
Yes. Okay. So I will not hopefully take too much time here. What I want to do here is to do a little bit of a deep dive into two slivers of our data to illustrate how we think machine learning would help us. I would like to start with these beautiful illustrations from Ramon y Cajal, which are as much art as they heralded an era of neuroscience. So it has been one of the longest quests in neuroscience has been to photograph the brain and to observe structures in minute detail and to understand what changes in a healthy situation versus disorder. One of the focus areas is and there are several levels of neuroscience, but one of the areas that is under intense investigation is to understand how neurons connect with each other and form these specialized structures called synapses, which are thought to be the business ends of neurons, where information gets, electrical information comes from one neuron on the presynaptic side, it gets converted into chemical information, which is then again detected on the postsynaptic side and converted into electrical information. And this process, the molecules that are involved here, the molecules that keep changing and the circuits that keep changing is thought to be the basis of learning and memory. So one of the ways in which in our lab, we look at these structures. So I must mention that these synapses are below the resolution of a typical light microscope. And so when electron microscopy came along, it was a big advancement to be able to observe the fine structure of these synapses and to understand how neurons communicate with each other and send information. In our lab, we developed tools and rigs to look at these synapses below the diffraction limit of a light microscope. And just quite simply to illustrate what we do here is to, instead of capturing one snapshot, we capture a few molecules in each imaging frame. You can do a Gaussian fitting to find out the XY coordinates. And then you build a so-called pointillist image from all these coordinates. How this works in real life is shown here, whereby you can have two things. One is, this is a tissue sample that is fixed, whereby there's a laser that comes on, activates a few molecules. And so because these molecules are not moving, we can iterate this over several thousand frames to get structural information. What you can also get is dynamics information, where if these molecules, so this is a live cell membrane, and each individual molecule is able to move around, you can then get a lot of information about their diffusion and dynamics, which is something I'm not going to talk about today. So using these tricks, you can go below the diffraction limit of a light microscope, and what you can see here is these are two proteins, one on the pre and the postsynaptic side, green and the red. It doesn't matter what the names of the proteins are here, but this is just to illustrate that we can look at these structures now finally using a light microscope. And I want to illustrate one ongoing project to understand what our hopes and aspirations are for machine learning. In one of these ongoing projects right now, it's a collaborative project where we are looking at these hair cells. These are specialized neurons, sensory neurons in the cochlea, which detect sound. And so you have fibers here which deflect with sound. They are connected to the auditory nerve fibers, which send information via these specialized synapses called ribbon synapses. These are present in the ear, and they're also present in the retina. And these use pretty much the same components as the synapses in our brain, but they're organized very differently. And so they're organized very differently. They also send information very differently, because when you have a small amount of noise, you have a few vesicles that fuse, that release the chemicals. Whereas if you have loud noise, you may have more vesicles that are fusing. And so they're typically designed to have tonotopic information that is getting transmitted. So our interest has been to model this structure using, which so far has been looked at only with electron microscopy, and to understand how this structure is built. So I'm going to show you these two cases whereby these are derived. These are these ribbon synapses. One of them is coming from a normal mouse, and the other is what we are seeking to do here is to compare mice which are born congenitally deaf. And these mice have all the structures intact of the ribbon synapse. It's just that these vesicles are not filled with a neurotransmitter chemical, which is why they cannot transmit. They fuse normally, but they have no information that is getting transmitted. So we decided to look at, rather Gayatri, a student in the lab, has been looking at these synapses. So we typically, what we do is we isolate these structures from the images. And what I'm showing you here are the postsynaptic side, which is the recipient of the information. And we can detect various receptor molecules. What we found, what we find here is that surprisingly several of these molecules seem to be organized to form a donut kind of shape. So Gayatri went about trying to understand how to quantify donuts. And what we found, interestingly, was that there seemed to be, so these are two different plots here. One of them is from mice that are very young, around 15 days after birth, which is when the onset of hearing is thought to happen. And there are mice which are a little older, 24 plus days after birth. What we find is that if we quantify the shape of the donut, there seems to be a transition that seems to be happening with the onset of hearing. In other words, the donut is less pronounced initially and becomes more pronounced as the mice age when they are getting auditory information. Surprisingly, this transformation doesn't seem to happen in these deaf mice, whereby most of the synapses that we seem to get here do not have this donut shape. There are other alterations as well, but I wanted to use this example to, and also other examples here from our data, whereby we can collect information on these on receptors which are on the postsynaptic side, which are detecting the information. And I'm sorry, this is not visible, but this is a Voronoi diagram over here, whereby we are trying to now also find out what is the degree of clustering of these molecules. And this is being done in collaboration with Vishnu Kabir as well as Prasad Perlikar at TIFR Hyderabad. What is interesting here is that several of these methods that we find ourselves using are already are being borrowed from astronomy, because it's these are similar kinds of data, similar kinds of clustering algorithms. The scale bar is different, it's in nanometers over here. But what our aspirations here are to, some of the data that I showed you, a lot of it requires manual curation. A student needs to look at these images, they need to crop them out, we need to rotate them in 3D, reorient them such that we can get the XY coordinates, map them onto a grid, and then get further information out of these synapses. So what we hope to do is that we hope that we can train machine learning algorithms to automatically identify these structures from our single molecule microscopy data. We also hope to increase, because this is such a labor intensive and at times subjective to the person who's doing this analysis, we hope to increase the n, our sample size, by a factor of at least 10 or more, so that we are actually looking at, instead of looking at a few hundreds, we are looking at few thousands of synapses, so that we can then realistically capture the variability that exists in different brain regions and in different conditions. So this is one. I wanted to move to the other end of the spectrum, where I am showing you a video from our lab. Now, these are two mice. This is this video has been put through a software that came out recently, which was actually developed for flies, but we can track mice and ask how they are behaving. So what we can do is, using this, we can quantify how these mice are coming into contact with each other, how much time they're spending in social contacts with each other. What these algorithms cannot do as yet is to do what we would typically have an undergrad do, which is to watch hours of these videos and discern, are they mating, are they having aggressive interactions, and more nuances of these. So we really hope that with advances in machine learning, we can do a little more automated analysis of these behaviors to be able to then use these to correlate the earlier structures of the synapses that I talked to you about in the brain regions with what is going wrong in the behaviors, let's say in certain neuropsychiatric conditions. So I think I'll end my talk here. And I guess since there are no questions, we can, I'll be happy to discuss later on. Sure. Yeah, what I meant is we are not doing any questions. Thanks.